how to organize and present your data and your figures in tables. To do the statistical analysis, you have to have an expert, and it's a topic that will take a long time to cover here, of all the different methodology to tabulate and analyze your data if you want it to be significant, and so on and so forth. So we're just talking about how to present, once you have the data analyzed, how to present it in figures and tables. So you, how do you know you have enough data for your article? Organizing your research data into figures and tables, like one of the participants mentioned. You start there, and most publications have 5 to 7 figures and possibly more now that you publish online. But when I was learning, I was told 5 figures was the max. Tables and figures, you should have 5 to 7. You don't want to overwhelm the reader with 10, 20 figures. It gets too heavy and too difficult to read, and perhaps you have enough for 2 articles then if you have so many data. So you, to make sure that your data kind of makes sense, present it to your colleagues, because that's what is going to help you. Present to your research group, figures, data, finding, presented to lab coworkers, because you know, you can have your raw data, your unfinished data, and then say, does this make sense? We are really immersed in our findings and in our experiments. When you ask others, it's much better to have a perspective that is not so close to your data. You can do poster presentations, and depending on the formality, you can include data from other people. You can present it in conferences, more formal. Usually the data is easy in a review format. You present, this is ways of presenting, you present an article. These are all pre-publication or can be done simultaneously to a publication. You can present a poster, you can present oral in a conferences, and it is still not public, not published. It's still new data, and then you submit it on a manuscript, on a paper, with original data and figures that only only come from your own research, and that is going to be the part that succeeds or stays in the literature forever, right? So you have to be careful to do it with original and new data. So how do you display your data? What type of data you have, tables and figures or pictures. The results should be presented so that they're easy to understand and support your main point. A lot of data points, results from 5,000 tests, must be summarized in a way that is easy to understand. And I will show you different examples for these choices. Try to use a variety of types of figures. Boring to have 6 identical types or graphs. Or you see articles that have a lot of tables. Usually are epidemiological studies that use a lot of tables, long tables, that it gets boring and it gets difficult for the reader to follow. So it's better to alternate. And typically you will have your table with your initial data. And then when you do further analysis, you can present graph bars, or you can present a continuous change on a figure, on a graph, on a curve. So combine it, make it interesting. Find new ways to present your data. I'll give you some examples. So this is a table that has the time and the sample was taken, the normal blood glucose levels, and a diabetic person. And yeah, it's a nice table, but you kind of spend, you know, it's like, yeah, so what is this telling me? So it's better to have a figure, and much faster to understand. So now you know that by breakfast, by the time it was taking in the morning, so breakfast, lunch, and dinner, diabetic blood glucose levels vary versus the normal. So it's easy, direct. You go, oh my God, yes, and then you move on, and the reader will be very happy with this kind of graph. Same here for antibiotic resistance, amoxicillin, many others that we cannot read, but it doesn't matter. You're just doing your controls, and then you're trying the resistance or amoxicillin plus CVS, and then you, very easy to understand, better than the table for sure. So that's what I'm trying to convey here. Again, this is raw data from a leishmaniasis paper that it had a lot of immune responses to leishmania. This is from Nicaragua. And then they found that the IL-10 is the one that changes because many others were equal to the asymptomatic and symptomatic. And then by analyzing, looking at your data, you decide to choose what's different rather than what's normal. You just say everything was the same, but this one, and then we came up with a graph. Well, not me in particular, but in my team, the active ACL for IL-10 levels was very different from asymptomatic and the controls. So you don't have to present all the data in graphs and figures. You only choose what is new and different to show the reader. And in the text, you will say, oh yes, this data, we analyze all the cytokines, and all of those showed similar levels from control to cases to symptomatic and asymptomatic people. But we found a big difference in the active ACL and IL-10, and then you present this graph. So that way you summarize in a very nice way what the reader should know instead of having to be bogged down with big tables of data that are not clear. You choose what's interesting and you put it on a graph, and the rest you just explain that it was not interesting, that it was the same as the controls or not very different. And I'm going to repeat this because I repeat all my information. See this figure legend, so nice under the figure. The figures and graphs have legends. The tables have titles, and we're going to see that more in detail. But this is, I'm going to take the opportunity. Figure 1, IL-10 produced by PBMCs in response to stimulation with Leishmania antigen. This is the title. And then peripheral blood mononuclear cells were collected from people with active atypical cutaneous leishmaniasis infection, people asymptomatic, and people non-infected control, and were stimulated with 2 milligrams of soluble leishmania antigens, and the IL-10 levels were measured by ELISA. This is your whole methodology. And I am sorry, you have to repeat it below the figures. This is how it is. If you see articles published, they all have figure legends where they tell you what they did. I know it sounds like repetition because you already put this in the material and methods, but for the reader that's not going to read your material methods and just results, and this figure is so nice, he or she are going to want to know how you did it. And there it is. And all figures have that kind of legend that explain exactly what you did. Okay, so that was a parenthesis. So be aware presenting a small amount of data in the graphs. Showing data in graphs can be dramatic and effective way to show an effect or a trend. But if your data set is too small, you can't say that you're seeing a potential or a trend. We're talking about trend or real effects here that we show in figures. So showing a graph could be misleading. You don't use a graph with small numbers, and here's the exaggeration of an article that had three mice, 1 didn't respond and 2 responded, or 10. Percentage of mice that responded to amoxicillin treatment, with 3 mice were treated with 0.5 milligrams of amoxicillin for 6 days. No, you don't do a graph. This is a little exaggerated, but if you have only 10 cases, a few cases, you don't do a whole graph. You just say out of the 10 studied, this was found. Unless you have multiple data for these 10 people, so then you'll do a chart. or it's also when you are presenting data that can be written in one text or one sentence. The comparison here, number of positive influenza and number of in different clinics with number of patients doesn't compare really well, and it doesn't tell you much, right, in a graph. It's better to have a table. So the figure present data of 486 patients and the influenza in different clinics with 24 of 446 were positive for influenza. So this is just the data that's showing, and it's very hard to compare 24 versus the 486 patients. It is a very pretty graph, but it's not necessary at all. You can just say in the text that these many patients were studied and then 24 were positive. That's it. You don't need such a beautiful graph to do such little info. So it should stand alone. Each table and figure be understood by itself. Should not have to read the text to understand it. And titles and descriptions should be enough. That's why I read the example of that nice figure that we saw earlier. When the reader sees a table or a graph or a figure, all the data to understand that table should be present. He should not have to go to material and methods to understand what you did and what are the results of the table. It should stand alone. So we have graphs, charts, pictures, and diagrams, and people are asking yesterday, what is the difference? A graph is a data in connected series that go linear. A chart is a data in separate series, like those three mice that didn't make sense. And a picture, those are more expensive to publish, and only if you must show a photo, let's say, of a laboratory immunofluorescence cells lighting up, or I don't know, a map, a photo of an area that you're studying. It has to be necessary for the results to show this photo, because it's not recommended if it's just to show pretty things. And a diagram is, you can make models to show your concepts, and I'm gonna do an example of that. So use the graph to show a trend or a pattern. Generally, a graph is not necessary when trends and relationships are not statistically significant. So that's when the analysis of your data is important. If a cause and effect relationship, so if it's a dependent variable, X is dependent from Y, then you do a graph. And for example, this graph with the glucose levels. Is dependent on time and dose, and it compares the normal versus the diabetic person. And this is another type of graph with the vaccine for diphtheria, and how, so it will be blue, and how the cases went down to almost zero in diphtheria, whooping cough, and measles vaccines that were applied over the years. So it's a continuum database, and then it gives you all the diseases that are present. It's a nice graph, a little hard to follow because the colors, but yes, if you want whooping cough, measles, scarlet fever, you can follow scarlet fever in the red rather easily. And it gives you a lot of years of data that very valuable. This is a funny kind of way of showing the survival of mice when treated with dengue 2. So survival and days post-infection, so 0, 5, 10, 15 days, these mice didn't make it. They died really fast. And then these survive with these particular mutations. And here the legend, I'm not gonna go through it again, explains what kind of mice you did, what kind of infection you used, how many particles of dengue you added, whereas the p-values, everything is done. So you see this and you understand perfectly what they did and what the results were. and what was the population of each experiment, how many mice they use. This is a figure with bars that it can give you a lot of information. Also, but it's not continuous. It's just from several different years, 2009 to 10, 10 to 11, 11 to 12, but it's not continuous, and it's different topics that are studied with concussions of football players, which is a big problem in this field, in the not our football, but the soccer, not a soccer or football that we all play, but what in the United States they play football. It's a different game, as you know. With helmets, and they clash constantly, and then head contact, how it affected, blunt sighting, body checking into head contact. Usually it's head contact, and it's been papers, articles saying how dangerous this is and how damaging it is for the person. They're trying to better the game, but it's not succeeding very well. A microarray data, which actually Stephen used to work a lot with this, of looking at RNA expression from thousands of genes. The red is upregulation, the green is downregulation. How do you present this? It's like, oh my God, okay. Hundreds of genes expressed, so you can do a table. It's choosing the most important ones, the genes that you want to show. These were upregulated for 30 minutes. Now these were downregulated for 30 minutes, and you have a lot of data. But actually 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So you chose 12 of the genes that were upregulated, and then you chose 1, 2, 3, 4, 5, 6, 7, 8 of downregulated. So this is a nice way to putting when you have too much information, but there are different things you want to show of a particular set of data. Like, for example, they are upregulated, and there are many different genes that are upregulated. So, oh, okay, so the big one is metabolism is really upregulated in here, etc. And then again, the figure legend, the expression, everything it was used is while the strain was growing, and how many 30 minutes at 37 degrees is amazing. That you are extracted the RNA, and then you determine it with the chip extension system, etc., etc. So everything is explained. You see this figure and you go, oh, this is what they did. It's fantastic. It's beautiful what they found. So this is another way of presenting it. This is downregulated genes, and this is upregulated genes. Maybe more practical, but you can have them directly in one graph. What's upregulated and what's downregulated, and in the time of exposure. So maybe not as beautiful, but more practical. You can show, and then you explain everything that you did to obtain this figure. So I'm just going to give you examples of figures, and this is when you absolutely have to have a photo of macrophages with dengue, infected with dengue. So this is beautiful, and it's actually from a colleague of ours that also teaches this workshop, and it ended up in the cover of that particular journal she published, I've forgotten which, because this was a spectacular figure that showed expression of genes of the growth of the dengue virus and expression of certain genes. Her name is Jennifer Kyle, and she's from Eva Harris' lab from many years ago. So this is when you can justify showing a picture, when it's absolutely necessary to show expression, growth, and a cell culture, and it's worth it. Survey maps also can help when you're covering a great area and you're gonna say what was studied. So this is in a survey in Boca del Toro in Panama, where the Panama Canal is, and all the studies they made. In the Smithsonian Institute that it had the Smithsonian Institute has a big research center in Panama, and they studied this area. It doesn't say what for, but it was a survey. Oh yeah, of different areas. Okay, well, we're not gonna dwell, just to show you where Panama is. So this is Central America. This here will be Colombia. And this here above Panama is Costa Rica. That's where I am at the moment, even though I work in San Francisco. I'm currently in my country of birth. Sorry, I lost myself there for a moment. This is a photo of a graph. This is a photo of an experiment of a gel, a protein gel. It's not very pretty. I think you can do better and present better gels, but this is very helpful. This will be the controls or the molecular weight controls. And this is the expression of the protein you want. Effect of dengue virus on protein translation in the presence and absence of an inhibitor. So this will have the inhibitor and says the doses of that you added of the inhibitor, and you can see how the, oh, sorry, these are the markers. This is the production of the protein and how it was inhibited. So it's relevant to show and explain what you did, and then you can say what was inhibited and what was not. Okay, and then this is the diagrams that you can make to show complex relationships. We're not going to read the diagram, it's very complex and it's hard to read, but you can explain like cluster 1, cluster 3, 2, cluster 3 of a particular effect that you're seeing, and that relationships usually in the discussion or at the end of results you say, this is how I think things relate. This is how I think this mutation affected this other mutation. This sequence compares to these other sequences, and you have a model to explain all the different interactions. This is a bit complex. Just an example of what you could do. And this is one of my favorite graphs of vaccines. It's hard to read. I don't know if they're worldwide. So when smallpox appeared back then in 1900s and now in 2010. So, so you see the effect of measles, which was big, and measles went down to this dot after vaccination. So it's very exciting to show, I like it. Rubella, this green one, and then vaccination, it basically disappeared. This is a good thing to show people that are resistant to get vaccinated for the COVID-19 or any other, how vaccines can be effective in reducing the presence of the disease in the world and how beneficial they can be. So I like this. I like this kind of figure that has colors and show drastically a change. So actually, I'm gonna carry this photo around and tell people that don't want to get vaccinated and show them what happened in the world after vaccines. No, to summarize, sorry, deviating too much. There are different types of presenting your data: graph, charts, pictures, gels, flow cytometry, that's very common, or diagrams that explain your complex or your relationships you want to make with your data. And all have figure legends, title, brief description of the experiment, and the table will have titles. So so here's just to break down title, one sentence identifying the main point of the figure, as you saw for the examples. The examples are more helpful. Brief experimental details, enough details so the reader can understand what was done to obtain the data in the figure, as the examples you saw, what experiments, how many, how units. Or you can do symbols, definition, this is a simple figure, antigen, present, control, and then you show difference in infection. And statistical numbers and p-values are usually welcome to be added when you have big databases that you're presenting. You show your numbers right there in the figure legend. When to use a table. Use a table when you have a lot of numerical values, but try not to pack too much information in the table, like I was saying, and don't include columns that have the same value throughout. If you did different data points, if you study different organisms and they all show the same, you don't have to have a table for that. You can do that information in the text and say, all this, we studied 10 microorganisms and all 10 were resistant to antibiotics. But if you study different antibiotics and they show differences in resistance, then you put the microorganisms and then the antibodies they were resistant to in a table. So tables are organized in columns and rows. Organize tables so that similar items read down and not across. So as I was saying, you put the organisms and then you do the similar items, what antibiotic they were resistant to, so you don't have to repeat the name of the organisms. The table has a title, it has footnotes, and look at the format in other articles as a guideline to how you're going to do your table. Again, that's what we're talking about Jane this morning, and other tools that help you choose your journal. It's going to have the kind of tables that they like, what size, what font, how big, how many tables and figures, and it will guide you in what they want. The more you do what the editors want in the journal, the higher the chances to be published. That's why we're trying to adapt to what their guidelines are, because we depend on them for them to publish our data. So here is a table, table title. It doesn't have legends, just a title, table, baseline differences in socio-demographic characteristics and treatment group in Ecuador, Panama, and Uruguay, 2005, 2006. So you put what's common on them to go down and what's different. So gender, male, female, what is the percentages? Very similar. So, well, ages show differences. So this is worth it for the table. When you see very little differences, sometimes you could eliminate it from the table and say, there was no difference between male and female and the response to amalgam or restorative treatment, and you don't have to have it. But since you have this other data in ages and different countries, maybe it's okay to have it there. But if you have too much data, you can eliminate this and say in the text, there was no difference between males and females in the response, but there were differences by age and countries. And the urban location, geographic location, if it's urban or rural, there was a difference or not. So that's how you try to group a table. So this is the characteristics that use the topic, the subject you study, and here is what you study and what are the difference show among them. Another example of a table. So this table looks like you have a table legend, but it's like a figure, but it's not. It's actually footnotes. This is a concussion with the football players in the United States I was talking about, and then the incidents of games, whatever. So you have explanations and asterisks. So here's a plus, here's an asterisk, indicates the p-value. Rates were calculated by concussion suspected, and you explain how the rates were calculated by asterisks. So when you're talking about the rate, you have an asterisk and explain it here. When you talk about suspected, you have a plus and you explain it here, suspected cases. So that's how you explain what happens in the table. This is not a very pretty table, but it shows all the little details you have to show for each point that you are presenting. Footnotes are brief explanations about the data, including exceptions found, like you study 100 patients, but 5 were not responsive. You can put 100 patients and put an asterisk, 5 were not responding or were an exception. Abbreviations, you have to explain them, and the statistics, the p-value. And do not write out information that belongs to do not write out information that belongs in the results. You don't write in the tables what you can explain in the text. So this is a simple table. Odd ratio of failures or alternatives. Oh, this is the same in Panama. And so you have B, results are insensitive to cluster. And then the p-value. This is A, and this is B. So you explain what group, country, age, and sex, and then results are insensitive to clustering. Anyway, this is just examples of tables, good tables and bad tables. And I'm gonna add a book that was published free by WHO. It's called From Robert Day on How to Write a Good Manuscript. We have the PDF of the book, and we can share with you in extra materials that will be placed with the talks for you to see. Okay, so how do I stop sharing?
At the top of your screen, there will be an option in red. There you go.
Thank you.
All right, no problem. So should we do a quick Q&A before before we move to? Yeah, so we are scheduled for a break at 11.
Okay.
And it's 10:30, so we have a few minutes to go for a few questions. And I see that Stephen has joined us. So please, if you want to join answering the Q&A now, Stephen, welcome. So there are some questions here about several topics. So one person is asking, what is the best way to show large amounts of data?
Well, I presented several examples. It depends on the kind of data you want to present. If it can be divided in different sections, it will be best. Or do you want to present all the data really, or do you want to present what's important? Like in the case of the leishmaniasis figure I showed, you have a lot of data, and all of data you choose those things that are relevant, that you show change, and then you represent that in a figure. But long tables with a lot of data are not encouraged, and they're boring for the reader. So out of the examples I showed, you can try to choose, depends on your topic. Yeah, you kind of choose what shows a difference from one group to the other, and only present that. You don't have, ethically, you do not have to present all the data in tables and figures. You can mention in the text, in material and methods, 4,000 topics, 4,000 points where samples were taken, out of which then they were analyzed for this and this and that other characteristics. And then you have 4,000 points of results with different studies done to them. You have to choose and summarize what is relevant, what's important, and only present that. And then the rest, you explain it in the results.
Yeah,
and
just a compliment. Oh, sorry. I didn't want to interrupt. Just a compliment to that is, like Maria Elena said, you want to present the main finds in your main figure, but if you are requested or if you want to show the entire data, what you can do, you can make a supplemental material in which you can put all your Excel file as a supplemental material in case someone is interested and you want to present that data, but usually you do that if you are requested or if you really want to add it. But then I would do separate as a supplemental material.
Good point. Many times the reviewers ask for that supplemental data that explain where you got the table from. And then that means that you needed to add more data and you summarized it too much, or they just want to know. And then you add it as a supplemental data. Thank you, Gregorio.
So next question is also, I guess, related to when you have a lot of data. So the question is there a limit on the number of rows and columns of the table to be presented?
There is not a specific limit. You see tables with a lot of columns and rows. It's just a matter of on making it clear for the reader. If there's tables that have a lot of data points, but there's just few, it's okay to have up to 10 plus or minuses or the data in a simple way. But if it is really a lot of data for each, it's going to be confusing for the reader. So it's your choice. Don't make overcrowded tables that are hard to follow. You're going to lose the reader and the editor, and they're going to lose interest.
Thank you.
I think I would just reinforce
go ahead.
What Maria Elena is saying is if you have more than 10 or 12 rows in a table, then consider breaking it into 2 tables or finding another way to present the data, right? People are much better at absorbing information if it's in a graphic format, so using colors and charts. So if you have a large table, either find a different way to present it or break it into multiple tables.
Good idea.
Thank you. Yeah, this was actually the next question from Leah, which is including color in the tables and graphs a good thing or a bad thing?
It depends on the journal, because some journals charge for color, and it's nicer if you have color. But some figures are just white and gray and shades of gray because they charge for color, or color doesn't represent well in the journal. So I would say to be conservative on the color, even though I presented very colorful figures. Those are allowed, but they have to be very intense, very good images for the color to translate in the publication. So if they are in shades of gray, might be less expensive and easier to publish.
I mean, I think I agree. The one thing is that more and more journals are online only now. And if the journal doesn't actually have a print version, then color there's no drawback or additional cost to it.
Of course.
So, and Maria Elena, I remember you and I learned if there is a print journal and they're charging for color, then to use shading.
Right. But of course, now we're going to move into publishing everything online. Although many journals still have a paper copy, the online versions will show colors very nicely.
Thank you. I'm going to make 2 questions as one, because they're all about guidelines. So one is, does data from the tables have to be included in the word count of the manuscript? And the other one from Shakuntala is, what is the guideline for supplementary material? I mean, how many pages? Sometimes people have very long supplementary materials. Thanks.
What was the first question?
If the words in the tables count as the word count.
The answer is no, sorry. I was just thinking about the word count is only for the text. So you can have the figures and tables, those are separate. To include supplemental, you don't want to excessively have supplemental. Those are submitted when you submit, not part of the paper, but annexes. that are submitted to, usually only if requested, or only if you see it's really, really necessary to present your data, or extra material like maps and things that you're not sure you should include in the journal, or that it will benefit the reader, the reviewer, the editor to understand better. And you include it as an annex or extra materials in the same submission. I don't encourage to present a lot. People that present a lot, those are not for the reader to see. This is just for the editor to make the decision. Extra materials are not going to go on the publication. So I don't think it matters a lot, but do not overwhelm the reviewer with a lot of extra materials. Usually they will ask, present more of this. We need to know where you get all this data from, and they will ask, and then you send all your database or more data.
Yeah, that's usually high impact fact journals. They would require some supplemental material, but it's like Marielena said, I would wait the reviewers to ask for more evidence than actually just showing everything at the same time, because you're going to overwhelm them and they may not be very happy about it. But of course, if they have questions about your work, they probably want you to send extended figures or raw data or something else, so it can be available to other people and they can actually evaluate your work from your main article. So there is no set guideline and how big can that be. It can be very long, very extensive, but it really depends what they're asking for you. Just don't overwhelm anyone in your first submission, for example. And of course, guidelines in general varies from journal to journal. So every time you go for a journal, you should check what the guidelines are. So it's the best way of approaching and deciding how you're going to format your article, which is actually also a little bit annoying because sometimes you want to submit from a journal, you want to go to another one, you have to reformat your paper almost entirely because the guidelines from each journal can be very different.
Thank you.
Thank you. A question from Hubert. Is there a standard table format for publication?
Is there what kind of table format?
A standard table format.
Each journal will have, yeah, well, standard is like the tables you see in the other journals, not overwhelmed of data and nice and clear. So what's the standard is what should go on a table. It should have a title, and it should have explained the data points, the statistical analysis with asterisk, footnotes on the table. That's the important guideline. Like Gregorio was saying, each journal will have a style, and it will tell you or will show you in different articles that were published, similar to yours or not, what kind of table they accept. And you should follow what journal you like when you're preparing your presentation, your manuscript, and you hope to publish in the journal. Follow that the guidelines and that pattern.
Thank you. Another question from Francis. So this is a longer one. Do I have to seek permission from the authors to cite a figure from an open access journal, say a figure illustrating a conceptual framework that I have used to analyze the study? Or is it okay to just cite the original source? If yes, what if I don't hear from the corresponding author? So I guess the question is, do I have to ask permission, or can I just cite when a figure was published in an open access journal?
I think you always have to cite, usually you have to ask the journal for permission, not the author. Once you publish, the rights for publication are from the journal. Open access or not open access, you always have to put the reference. You always cite it. As for permission, I don't know if Gregorio and Stephen have more experience with this, but you always have to have a footnote that said this graph, this table, this data was published in this particular article with permission. So we publish in this data from this article with permission. Usually you have to ask the journal for permission.
Yeah, I mean, Maria Elena, you're exactly right. And that's actually a very important point, because to do otherwise legally can be considered stealing. And it can also lead journals to refuse any further opportunities to publish.
Thank you.
Thank you. So one last question from Safa. When writing rare case reports, do we need to add supporting information and photos?
Supporting information always. I don't know what she means by that, but photos, I'm not sure. Probably one photo will be accepted. If it's really rare and different and you want to show a new syndrome, a new manifestation that's never been seen, try to limit the photos, maybe just one. But yes, I think it will be helpful.
Yes, I just wanted to add about the last question about the quoting citation. It's very important for the open access to check the license. It's normally the Creative Commons, and there are several license types. You can always use that, but the license types are different. And sometimes you can directly reuse it if it's non-commercial, and sometimes you can just cite it. And if you want to avoid some complications, whether from a journal or from the research group, just check the explicit type of a license. It's quite easy, just Google that. And if you Google that, then you can avoid most of the problems, because normally if you have an open access information, it's licensed under Creative Commons. So that was what I wanted to add. And it's important because our group had some problems with the junior researchers who haven't checked the license and they cited it in the wrong way. It wasn't a big problem, but you can avoid that very easily if you just check the Creative Commons type, which the authors or the journal have given the publication.
Very good, Lana, thank you. See, you can run into problems if you don't check with the licensing of the journal.